And welcome back to our last video in chapter 11. So the last thing we're going to talk about are how to identify mutants and other mechanisms of changing our DNA. So other ways that we can mutate. So we're talking about chemical mutations here, and then we'll talk about how some bacteria can transfer their DNA to produce new characteristics in other cells at the very end, okay? So mutations are rare in general, and the reason for that is because we have so many excellent mechanisms to fix mutations. And mutants are difficult to isolate, but there are two basic ways that we do isolate mutations or mutants. One of those mechanisms is known as direct selection. So it's exactly what you think. When we have a test tube filled with bacteria, you don't see any mutants within that test tube. In fact, you don't see the cells. What you're seeing is a cloudy environment made from all the material that the bacteria produce. And so what we do to identify a mutant is we're going to use a medium that only the mutant can grow on, and that would be a direct selection. So this is often how we find mutants that are antibiotic resistant. So why would we be looking for antibiotic resistant mutants, you might ask? Well, there's a lot of importance in isolating antibiotic-resistant strains of bacteria so that we can then find new antibiotics that will kill them off. So that's what a lot of scientists do. That's their job. And so in this direct selection, we're looking for cells that are resistant to streptomycin, which is an antibiotic. All cells are going to be able to grow on a medium without streptomycin, but they're not going to be differently colored. So if this is a E. coli, okay? E. coli look like E. coli no matter what. They're not going to change color here. And so this isn't going to help you. But if you plate it also on a medium that contains streptomycin, only those cells that are streptomycin resistant are going to be able to grow. And that's direct selection. Another mechanism, which is a little different, is called indirect selection. So in this case, we are actually selecting organisms that don't grow, which sounds really weird. So this is a really good mechanism for finding auxotrophic bacteria. An auxotroph is a cell, or is a bacterial strain, that has mutated from the original parents that had certain abilities to produce certain enzymes. So auxotrophs are organisms that have to have special amino acids or nutrients to be able to grow, whereas their prototrophic parents could grow without them because they could produce those amino acids. Auxotrophs don't have certain enzymes, and so they can't produce those amino acids. So why do we want to find auxotrophs? Why would we care? Okay, so the prototroph can grow, auxotrophs can't, so that means that they're not going to be able to cause disease. Well, we need auxotrophs so that we can test chemicals. So remember we talked about mutagens, and a lot of chemicals can be mutagens, and mutagens can be carcinogenic. So there's always inventors producing new chemicals. These chemicals can range from, something that cleans up, an environment, to something you might spray in your hair to hold your hair in place, to something you might put on your skin to help your skin look more healthy. So we're constantly producing chemicals that we're trying to sell for different purposes. And we have to test these chemicals to make sure that they're not dangerous before we sell them. And so auxotrophs are used in testing purposes. And so so here's how we find our auxotrophs first. we use a process called replica plating. So in replica plating, we're going to have bacteria grow in a complete nutrient medium. And we're going to then take a sterile velvet cloth. We're going to place the sterile velvet cloth, press it into the medium containing all the different colonies. We're going to take that sterile velvet cloth and we're going to press it into a medium that contains whatever amino acid that the auxotroph needs to grow. And then we place it in a medium that lacks that amino acid. And so we now have a replica, 2 replicas of this original plate, right? These are going to grow, set into an incubator, and grow for about 48 hours. And then we're going to look at the results. And what we should see is an identical replica of the original parent, because that's where the colonies were. But anywhere that we have mutants on the medium containing histidine, then we should have no colonies found in the one that lacks the histidine. And so right here we see that there's no colony, which means we know that this one colony here is an auxotrophic mutant. We can pick this colony up and we can grow it, and then we can use it for different testing purposes. And so what is the testing purposes that we're looking at? It's called the Ames test. The Ames test identifies the mutagenic properties of chemicals. So when a chemical is when someone is trying to put a chemical on the market, they have to first test to make sure that that chemical is not going to be considered a mutagen. And if it is a mutagen, if it's mutagenic, then it's more likely to be carcinogenic. And the more mutagenic it is, the more carcinogenic likely it is. So mutagens aren't all carcinogens, but if it's a very strong mutagen, it's more likely to be a carcinogen. And so we use the Ames test because it allows us to test to see if a chemical is actually mutagenic. And so in this test, we typically use salmonella, an auxotrophic salmonella strain. The salmonella requires histidine. So we've got it from that replica plating. We grew it. We have a lot of salmonella that are histidine dependent. We then grow that in a rat liver extract. Then we add to the experimental group a possible mutagen. And the other group, so whatever chemical we're testing, we would just add water, all right? We then plate the bacteria onto a medium that lacks histidine. The control group we're plating because there's always that potential for bacteria to mutate on their own. And because bacteria grow so rapidly, mutations occur more rapidly in bacteria. So we're looking at the control and comparing it to the experimental plate. And so if the control here shows 2 different colonies formed, if this plate only had 1 or 2 colonies, we'd know that the chemical is not really a mutagen. But if there's a lot of colonies formed, then we know that this chemical is actually a mutagen. And the more colonies we have, the more mutagenic it is. And that's how we can test chemicals using those auxotrophs. And that's why we need auxotrophic bacteria. The very last thing we're going to talk about is other mechanisms for transferring DNA. So we talked about, or not mechanisms, other ways that genes can be altered, or that cells can be altered. So we talked about mutations altering genes, which that can happen, but we also bacteria don't have sex. So there are different mechanisms that bacteria use to acquire traits from other cells of the same generation. And so there are three basic mechanisms that we talk about. We talk about transformation, transduction, and conjugation. These are all forms of horizontal gene transfer. So vertical gene transfers from parent to offspring when binary fission occurs. But horizontal gene transfer is the mechanism of transferring information from one prokaryotic cell to another prokaryotic cell in the same generation. And the really interesting thing here is that the prokaryotic cells don't have to be like cells. So you don't have to have 2 E. coli cells or 2 Salmonella cells. You can have a Yersinia cell and a Streptococcus cell, and they can share information. Transformation we've talked about a bit when we talked about Frederick Griffith. Transformation occurs when a competent cell takes up DNA that is considered naked DNA, and the naked DNA came from the death of another cell. So you have one cell that dies in a solution, the DNA leaks out, another cell in that same solution can pick up that DNA. Transduction we also talked about in chapter, I think it was 6, where we talked about viruses. And transduction occurs when a bacterial virus or a bacteriophage transfers genes from one cell to another cell. And then we have conjugation. This is the closest thing to what we would call bacterial sex. In this case, we have 2 cells of the same generation that one cell has a plasmid that contains a conjugation pilus gene, and it produces a conjugation pilus that attaches the 2 cells together, and then that plasmid can replicate and move into the new cell. And so here are the 3 mechanisms. Here we have transformation. This cell leaks out information. Okay, actually you're not really seeing that. You're seeing up here, a cell up here dies, leaks out information. That information is taken in by another cell, and then that cell incorporates that information into its DNA, and it's been transformed. Here we have transduction, where a bacteriophage takes up some DNA from some cell out here, but it takes up the genetic DNA of the cell versus the viral DNA. And then it incorporates or it infects a new cell with that bacterial DNA. That bacterial DNA then is incorporated into the cell, and now you have a new recombinant cell. And then this is conjugation. We have a plasmid that carries information to produce the sex pilus here. The plasmid so the cell produces the sex pilus, they attach, this plasmid replicates, and one of the plasmids moves over here, the other one stays here. For transformation to occur, cells have to be competent. So a competent cell is a cell that is able to take up DNA from an environment. There are certain types of cells that are not competent naturally, but there are some cells that are naturally competent. An example of a cell that is not naturally competent that we love to use all the time is E. coli. E. coli are not competent cells, so we can make them competent by shocking them. So we basically put an electrode or an electric impulse into the solution that the E. coli are in, and that stimulates the cell's wall to break apart a little bit, and that allows DNA to leak into the cell. And this transformation can actually allow cells to become pathogenic. Transduction then, genes are transferred from one cell to another cell by a bacterial virus or a bacteriophage. 2 types of transduction that we talked about before: generalized transduction, where some of the host DNA is packaged up randomly, and then specialized transduction occurs when a piece of the host DNA is taken up, the specific information that we care about is taken up. And so it's usually through or from the lysogenic cycle, the bacterial virus will break off and it will grab on the DNA that's right next to it, pulling it out as well. And then we have conjugation. Conjugation is the transfer of DNA through a structure called a sex pelus. During conjugation, we typically have two types of cells. We have a plus cell and a minus cell. The plus cell then transfers a plasmid to the minus cell, and then both cells become F plus cells. So the F stands for fertility factor. Okay. And so here you can see an F plus cell. Here's the F minus cell. There's that sex pelus that's formed. The plasmid then replicates, and in the end we have two F plus cells. This is the end of, oh, I lied, one last thing, transposons. Transposons are genes that we've often called jumping genes. They move from one area of DNA to another area of DNA. They can move on the same chromosome. They can also move from one chromosome to a totally different chromosome. And when they do this, they can cause problems for cells, because the cells, if that transposon jumps in between some type of a gene, that gene no longer will function properly. So it can cause problems with functionality, but it also can produce certain types of characteristics. If a transposon jumps from one chromosome to a totally different chromosome, it can actually cause the new chromosome to have certain characteristics that it didn't have prior to. And that is the end of our video, or our lecture. So I'm going to stop here, and I'm going to upload this, and we'll be having, and so I'll post it shortly for you, okay? Bye.